it's not just making a cell for the making of a cell. It's actually, I like the idea that we're making models that will one day unlock the mystery of the disease process that and lead to therapeutic intervention. MorPhiC stands for molecular phenotyping of null alleles in cells.
MorPhiC is this new initiative, consortium, and the goal of MorPhiC is to use human-based cellular models to get at protein coding gene function. And we do this by creating null alleles, knocking out genes in iPS cells, and then differentiating these cells and using different strategies to phenotype the consequences of the loss of those genes. The reason we want to knock out genes in human cells is to get at the function of these genes. So there are approximately 20,000 protein coding genes in the human body, and many of them we still don't know what their function is, or one gene could have multiple functions depending on the context that we study it.
We don't know what the function of most genes are in cells. We have lots of different assays that we can use to look at what the effect of removing that gene is on the cell. So there's two goals. One is to collect data on gene knockouts, and then to take all of that data, which is a huge amount of data, and try to synthesize and understand what that data is telling us. So that's the data generation aspect of MorPhiC. And that'll go on for years, and the data can be reused and reanalyzed for many years to come. But the second goal would be to create a permanent resource of cells that each contain a knockout, which can be distributed to individuals within the scientific community who are interested in that.
My collaborator and colleague in MorPhiC is Bill Scarnes, and he is his lab is the one that engineers these null alleles within undifferentiated human iPS cells. And then once he's confirmed their actual nulls, then he'll pass them to my lab. And we take these undifferentiated cells and then apply different differentiation strategies.
Working with cells in culture, it's like Miracle-Gro on a plant. They are grown at 37 degrees usually in an incubator, and they need carbon dioxide so the pH of the media they're growing in is buffered. There's a lot of work going into trying to create mini brains from these cells, organoids of various organ systems. So it's only going to get more and more sophisticated as we go along.
Part of a collaboration is it's really important to trust that individual, follow up on what you say you're going to do in a collaboration. And the ultimate goal is joint success, leveraging complementary capabilities in addressing scientific questions.
It's great to have colleagues around down the hall who are interested in the same problems that you are. And so JAX does a good job of focusing on certain areas of biology and recruiting scientists in those areas. I can't imagine working anyplace else other than a laboratory, because a lab is a place where you solve problems. And so every day you come in, it's a different problem, so it stays interesting. So the job has always been interesting. You always want to know what's around the corner. I want to be hands-on. I want to be making those discoveries. That's the greatest thing, and it doesn't happen very often, where you come in and you've actually discovered something important. The thrill you get from that is just incredible.